All right. Thanks very much, Luis, for the kind introduction, and thanks to the organizers also for giving me the opportunity to present some of our data on NS5A, NS5B, mechanisms of drug action and drug resistance, and to talk also a little bit resistance in general terms. I'd like to start out with some principles in the development of resistance. So there are two major factors that we need to consider here, and that drives the development of mutations and in turn also selection of resistance. And this is first of all rapid replication, the production of an enormous amount of viral particles, and secondly the error-prone nature of the RNA-dependent RNA polymerase of the hepatitis C virus. So these two factors are really key in a better understanding of the emergence of resistance. And as you can see on this slide here, what happens in a clinical setting here, an individual is infected with the hepatitis C virus. Usually there are millions and billions of different variants, viral variants that sometimes differ just by a single amino acid. And once therapy is started, the sensitive viruses here indicated in green, they cannot further replicate, die eventually. And those viruses that can survive under these conditions, like the orange one here, they eventually outgrow the population, and all that is then associated with a rebound and viral breakthrough eventually. So that was theory, and this is a similar slide you've seen from Mark before. This is a dual drug regimen, protease inhibitor and NS5A inhibitor. Small group, 7 subjects here, and you can basically see with these two DAAs there is relapse or rebound to baseline levels in each single case, and that is associated also or correlates with the emergence of those variants here that are associated with resistance to NS5A inhibitors. So these are usual suspects here, Y93, L31, and with regards to NS3 protease inhibitors, mutations at position R155 and D168. So this is a resistance map, so clinically relevant NS3 for a resistance mutations identified in vivo, and many of them can be selected also in the replicon system. And you can see also really the 155 and 168 almost, they confer decreased susceptibility to literally any of these available protease inhibitors. Just one word regarding the mechanism of resistance here. You have to understand that there's a substrate envelope that basically covers the average substrate that would bind to the active site. And if certain amino acid residues would stick out these substrate envelope, they can be rapidly mutated without having any penalty on the substrate that is actually bound to the protease. And in the case of 155 and 168, and that was demonstrated also by crystal structures published by Celia Schiffer's lab, these mutations, they stick out of these substrate envelope. So that's why the fitness penalty is often really minor, and those mutations can linger around. With regards to NS5A inhibitors also, there are a bunch of mutations here, and Mark alluded to this. Some of them really confer cross-resistance to the entire class. Position 93, this is definitely an example here. For NS5B inhibitors, we really need to distinguish between nucleotide analog inhibitors and non-nucleotide analog inhibitors. And you can see for the non-nucleoside inhibitors, there are also a bunch of mutations here, and there are even more. It really depends on the particular class. And what really is evident that there is only a single mutation, and I come to that. It's not really true. There are also some other minor mutations that may be important in regard to resistance to nucleotide analogs, but this is the classic signature mutation S282T that confers resistance to nucleotide analog NS5B inhibitors, and specifically to those that have the 2C methyl modification in the sugar moiety. And I come to that regarding the mechanism a little bit later. At this point, it is only important to recognize that there is indeed a very high barrier to the development of resistance with regards to the S282T mutation, and most other mutations that I just mentioned here regarding NS3, NS5A, or the non-nukes, they can be relatively rapidly be selected in vitro and in vivo in the context of monotherapy. So the big question is why also? Why are certain mutations rapidly selected while others are hard to select? And the S282T mutation is an example for a mutation that is difficult to select for, and there are indeed just very few examples in the clinic that this mutation shows up. And this was presented here by Mike Miller at last year's HIV/hepatitis C drug resistance meeting in Berlin. And so there's one subject here on sofosbuvir monotherapy and one subject here on sofosbuvir NS5A inhibitor dual therapy. Here are some other mutations, and I just alluded to those mutations here, L159F. So if you really do deep sequencing, and the FDI people did this, you can actually identify those mutations here, and some of them, they already exist at baseline. The clinical relevance of these mutations is not quite clear at this point. However, studies in cell-free assays with NS5B as well as in the replicon system have shown that there's decreased susceptibility to sofosbuvir and related compounds. Now, this is an example here of the one of the patients that I just mentioned. So sofosbuvir in the context of monotherapy resulted in a rapid decline in viral load, and then in the follow-up there was relapse, and they genotyped the virus. They identified S282T at this point, and then later on not anymore, and instead the population was outgrown by wild type again. So they did a retreatment here, and it was actually successful. And the retreatment contained, on top of sofosbuvir, ribavirin, and the period of treatment, or in this case, one was not even longer. So this is a major difference also to HIV, where in HIV there are reservoirs, and eventually those reservoirs, they may still harbor resistant variants. So retreating with certain drugs in HIV is just a no-no, and that's why resistance testing in that regard is also extremely important. Now, here it seems to work quite well, at least with sofosbuvir, and one of the reasons is actually can be seen here in this slide. You see that S282T, there's only a small window where you can actually detect S282T. A little bit later, a week or so, it's just wild type. And the reason for that is S282T, that variant, is severely compromised with regards to viral fitness. So that's why the penalty on the virus is extremely high. S282T would disappear and wild type would take over unless there's continuous drug pressure. So, but fitness, we thought, may not be the only reason that really determines whether a mutation shows up or not. There are two parameters, and the genetic barrier, so defined here as the number of nucleotide changes, also the nature of nucleotide changes, may play an important role, and I come to this in a minute. Viral fitness, I already alluded to this. This is a parameter that is, of course, extremely important, and it is often mentioned in this regard. Now, let's have a look at the nature of the mutation. So a couple of years ago, we published this paper here. We looked just at untreated patients and looked at the mutational spectrum, and we identified a bias towards so-called transition mutations. And these are purine to purine, pyrimidine to pyrimidine mutations, versus transversions, and that would be purine to pyrimidine or pyrimidine to purine. So if you wish, from one ring to two rings or two rings to one ring. And there's a huge difference. It appears as if transitions really occur at a much higher frequency, 2 orders of magnitude, 1 to 2 orders of magnitude. What does that mean with regards to the emergence of resistance? So if you look at S282T, it requires a G to C change, and that is a purine to pyrimidine change, so that is a transversion. And we think this is one of the reasons why this mutation is hardly generated. And in that paper, we also provided biochemical data for that. Transition mutations, they require GU or UG mismatches at the active site of the polymerase, and these are easily accommodated by NS5B. So that is a mutation that is easily generated, and that's why transition mutations are in turn easily generated. For transversions, you need other mismatches, G mismatches, for instance, and so forth, and these are hard to accommodate by the polymerase, and that's why they are not frequently generated. Another example in the literature is here the number of mutations. So R155K is often frequently detected in genotype 1A patients, and it's just a single transition, G to A. So it is rapidly generated, easily generated by the NS5B polymerase. While I think in genotype 1B, there are 2 changes required, and I think this basically never detected or extremely rarely. And it's not only the number, but it's also one transition and a transversion. So that is a genotype that is difficult to generate, and we would argue the genetic barrier in this case is extremely high. So it would probably follow the order, the highest genetic barrier is here, then second here, and then transitions here. So to better understand the contribution of genetic barrier versus fitness, this is an analogy here to chemical reaction, and you may remember this from a couple of years ago, in my case maybe 30 or so. So this is the starting material here, and you always need, in order to generate the product, you need to overcome a certain activation energy that can be lowered by catalysis, by heating up the starting material, and so forth. But if the activation energy is low, in principle, you rapidly generate the products, okay? And that is the analogy to transition mutations. Transition mutations are easily generated by NS5B, so you rapidly generate transition mutations. However, that doesn't mean that these transition mutations are also stable. For chemical reactions, same thing. It does not mean that these products are stable. So the other pathway is a higher activation energy, and the analogy would be a transversion. So you hardly generate the transversion. However, once generated, these transversions, they could be really stable, and in the case of viruses, they could confer high levels of resistance, and they could theoretically
linger around for long periods of time. In terms of the stability or fitness, I do think, and then there's evidence for this, that NS5A mutations in fact do linger around quite a bit, while the S282T mutation that I just mentioned disappears rapidly. So this would be the fitness argument here, and this is the genetic barrier argument here. So both of them need to be separated. Now, the question is now in terms of the mechanism, why is there a fitness penalty with S282T? And this is a recent structure here of a ternary complex, recently published in Science by Pharmacet/Gilead people. And you can see here at the active site there's sofosbuvir. It's actually crystallized as a diphosphate form. You can see that S282 is in close proximity to sofosbuvir. So it's not the mutant that they crystallized, right? But you can envision that the mutant, if there is an additional methyl group here with threonine, that there could be a steric clash also with the natural nucleotide, and that may prevent binding and or incorporation of the natural nucleotide as well. So there's a little bit of a penalty, a fitness penalty, because that resistance conferring mutation is in close proximity to the active site. Okay. Now, in terms of mechanism of resistance, here's the methyl group that sticks out. And one can imagine that with a threonine, the steric clash that I just alluded to might be even more severe, so sofosbuvir is not incorporated. There's no direct evidence for this, and I would like to refer to some work that we did in my lab. Anupriya Kulkarni did this, and she also has a poster here. So if you look at S282 in the context of NS5B, this is adjacent to G283, which interacts with the 2' hydroxyl of the templated nucleotide. Okay? So this is the RNA template here. And this is the incoming nucleotide. So S282 and G283 forms a ring-like, perhaps rigid structure here with the RNA template and the incoming nucleotide. And that may change if you have the 2' C-methylated compound here and the resistance conferring mutation. There could be a steric clash that may prevent also base pairing here, and the nucleotide is not efficiently bound and or incorporated. Now, we did one experiment to actually prove this, or to get better evidence for this hypothesis. And this is, we basically used a DNA residue at this position. So we are disrupting now the interaction to this highly conserved G283. And in doing so, we generate a bit more flexibility in here, and that sort of provides a relief here to the steric clash between the nucleotide or the inhibitor and the resistance conferring mutation. So in other words, the nucleotide can be accommodated under these conditions. And in fact, if we use a DNA template instead of an RNA template, we don't see any resistance. So that is pretty good evidence, we think, for a steric clash between sofosbuvir or related compounds and S282T. Now, the last couple of minutes, I will talk about NS5A. And as Volker pointed out, there are two different dimer structures that are published. Let's refer to this structure here as an open structure, or clam-like structure. And let's refer to this structure here as a closed structure. So these are structures that are published, and we the authors of this structure here, of this paper here, they argued, okay, this is a cleft here that would facilitate binding of a double-stranded RNA, while in this structure here, there's no evidence for such a cleft. So the idea was that in this structure, RNA could be accommodated, but not here. What we have done is we used exactly the protocol for expression and purification of these proteins according to the first paper and according to the second paper. And what we found is that only if we use the protocol for the first paper, the Tellenhusen paper, we actually do see RNA binding. If we use the protocol for the love structure, we do not see RNA binding. So there seems to be a correlation. What we don't know at this point with the biochemical data is this really indicative for the existence of this particular structure, and is this result indicative of this particular structure here? And to look at this a bit more closely, we developed a FRET-based assay, and what we see is there are different FRET values generated depending on the type of purification that we have chosen. So it makes a lot of sense that the open structure shows a lower FRET as compared to the closed structure that shows a higher FRET. Now, we did also site-directed mutagenesis to further confirm this. So we looked first at the open structure, mutated residues that are seen at the interface of the dimer, and what we see is then that RNA binding is compromised, and we see a slight increase in FRET. So that is indicative for a conformational change towards the closed structure. We cannot exclude also the extent of monomeric structures. We did the similar experiment here with the closed structure, picked another pair of important residues at the interface, and we see now that RNA binding increases and the FRET signal decreases. So this is the opposite switch from a closed structure to an open structure. So now, regarding resistance-conferring mutations and inhibitor binding, all of these mutations are clustered here in close proximity to the site that is attached to the membrane. And Volker mentioned also that there are a lot of models proposed that would help to better understand how inhibitors bind. And these inhibitors may bind in a symmetric way, in an asymmetric way. So there are a lot of models out there. I do think we really need to aim at structures to see what is really going on. And at this point, we cannot exclude also that multiple binding modes indeed occur. So in conclusions here, just for NS5B and NS5A, so there are high barriers to the development of resistance to nucleotide analogs. And in terms of the mechanism here, there's strong evidence that S282T causes a steric clash with sofosbuvir and related compounds, specifically at the 2'-C-methyl moiety. With regards to NS5A, low barriers of resistance, it is probably associated with the fact that those mutations may not be sufficient at an individual basis, and multiple mutations are required. The other argument would be NS5A is not an enzyme, which really doesn't facilitate also its study, and these mutations, they may occur also at positions that are not important, and that may help to explain why they linger around for longer periods of time. I really think that we need to get a better understanding of the conformational dynamics and plasticity of that protein, and that really complicates current studies of mechanisms of
of action and resistance.
So I would like to thank everyone in my lab who was involved in these studies, and first and foremost for the NS5A project, Nishal Isor. He's also presenting a poster here, and Anupriya Kulkarni, she's presenting the NS5B poster. Megan Powdrill did the work regarding the genetic barriers of NS5B, and all three trainees were actually supported by the NCRTP, so we are grateful for that support, and funding of this project was provided by the CIHR and some companies, and here our collaborators, Dr. Kosa and Dr. Russell. Thanks very much for your attention.
Okay, thank you, Matthias. If we have any questions for this work, perhaps I can start with one while we're waiting. Do you think that these two conformations of 5A are having different functions, or that it's a conformational switch in the same function?
So I do think at this point it is extremely difficult to correlate structure to any function. That is the challenge right now, and that's the aim also of our studies. The only function we could really ascribe to one conformation, this is RNA binding. How that translates into replication, we don't know at this point.
That was pretty much going to be my question, actually, is how have you looked at if you really impair the RNA binding? So I mean, with that Y93 mutant, you saw a slight increase in FRET, and so it looked and a little decreased RNA binding. But can you put that in and look at actual replication beyond that and see? Because I would expect that would actually have quite a significant fitness effect, and it doesn't seem to by the fact that it persists for so long.
Yeah, so that we haven't introduced these mutations now in cell culture-based assays, so replicon or virus. It would be interesting to see what the impact on the various functions, on the various different stages within the life cycle of hepatitis C would be. It is conceivable that these mutations may affect replication and/or assembly in a specific manner. We don't know.
Do you know if anyone's ever looked, like most people have focused on looking at the resistant variants and whether that affects outcome, but has anyone looked at viral kinetics in patients with those more susceptible variants that are more susceptible to binding?
Not that I know of. I mean, we certainly start to, and this is the beginning with 112 and these guys, we certainly start to introduce these mutations in a strategic way that we just block a specific parameter, let's say formation of conformation 2 or formation of conformation 1. And with that, we hope that those mutations still are viable and may give some insight into these type of mechanisms. So, thanks.
Okay, thank you.